बिस्मिल्लाहिर्रहमानिर्रहीम। आज के लेक्चर में हम पढ़ेंगे म्यूटेशन एंड इट्स टाइप्स। व्हाट इज म्यूटेशन एंड व्हाट आर देयर टाइप्स। म्यूटेशन क्या है और इसकी अक़साम क्या है। म्यूटेशन की डिफिनेशन इस तरह है, चेंज एंड सीक्वेंस आर स्ट्रक्चर ऑफ जेनेटिक मटेरियल। जेनेटिक मटेरियल की स्ट्रक्चर और सीक्वेंस में तब्दीली आ जाती है। Those factors which cause mutation, there are some factors responsible for causing mutation, which is known as mutagen. Mutagen are of two types. The first one is the physical mutagen. The physical mutagen, for example, X-rays and UV rays. These are the physical mutagen. X-ray physically attack on the body, enter to the bodies, affect the genetic material sequence and cause mutation. Same is the case of UV rays. Ultraviolet rays also strike with the body, with the plant body, with the animal body and create mutation, cause mutation. The second one is the chemical mutagen. The chemical, those chemical which cause mutation is known as chemical mutagen. For example, if we consider nicotine, the nicotine attack on the body's lung cells where they cause different type of mutation. There are other many types of chemical which cause mutation and it is chemical mutagen. Now comes to the types of mutation. Types of mutation on the basis of origin, that where the mutation process occur. We study the origin of mutation, the place where mutation occur. There are two types of mutation on the basis of origin. Number first is the germline mutation. The germline mutation, those mutations which occur in the germ cell. For example, the mutation occur in ova or in sperm cell. This is called germline mutation and the germ cell. And this type of mutation, the main important about the germline mutation, that the germline mutation run and the upstream. For example, if we consider sickle cell anemia, which transfer from upstream to upstream and run in the family. The mutation which occur first time in a cell or on DNA or new cell, this type of mutation is called de novo. Novo means new. New mutation. First time occurring mutation is known as de novo. The second type of mutation on the basis of cell or origin, this is known as somatic mutation. Somatic mutation, the mutation occur in somatic cell, in body cell. For example, cancer is a disease which occur in the body cell. This disease cannot run in the family and the offspring. This is the main important about the somatic mutation. The somatic mutation not run in the family. Now, types of mutation on the basis of how occur. Mutation किस तरह वाक़ हो जाते हैं? How. There are two types of mutation on the basis of how. The number first is spontaneous mutation. The spontaneous mutation are those mutations which occur naturally. How naturally occurring? For example, the body enzyme not function properly. They lost their function, due to which the mismatching occur of nucleotide mismatching occur. Due to this mismatching of nucleotides, the mutation occur, and this mutation naturally occur. That is why called spontaneous mutation. The second one is the inducible mutation. The inducible mutation, those mutation which occur due to external factor. For example, if we consider the physical mutagen or the chemical mutagen, both are responsible for induced mutation. Types of mutation on the basis of where the mutation types on the basis of where the mutation occur, the finds of mutation. They have two types. Number first is DNA mutation, which is also known as gene mutation and also known as point mutation. On the basis of single nucleotide, we consider this example on the basis of single nucleotide. That is why called point mutation, which occur in a particular point. They have further three types. Number first is deletion. The deletion, for example, consider a sequence which having 3 triplet and 9 codon. The A, C, T, adenine, cytosine, thymine, guanine, adenine, thymine, cytosine, guanine, and adenine. For example, due to some reason, due to physical mutagen or chemical mutagen, this one lasts. After the last, they become A, C, T, G, T, C, G, A. This sequence without A. This type of mutation is called deletion. This type of mutation is called deletion. The second one is the substitution. Consider this sequence. For example, if we have this sequence is cytosine, guanine, thymine, adenine, cytosine, guanine, thymine, adenine, and cytosine. In this type of sequence, if the guanine come and replace the cytosine, and this place, the G place and C out, this type of mutation is called substitution. There, cytosine is substituted by guanine. The third one is the insertion. For example, if we have a sequence of cytosine, thymine, adenine, guanine, cytosine, thymine, guanine, cytosine, and adenine. On the place of cytosine, if another one is added to this sequence will become cytosine, thymine, adenine, guanine, cytosine, adenine, thymine, cytosine, guanine, and cytosine and adenine. Extra one is added to this sequence. This type of mutation is called insertion. The second type on the basis of here is the chromosomal aberration. The chromosomal aberration have further two types. The one is chain and structure. This type of mutation, chain occur and structure. And the second one, chain and number, their chain occur and number of chromosomes. Chain and structure, consider chain and structure. The chain and structure have further four types. The first one is the deletion, the second one is the insertion, the third one is the translocation, and the fourth one is the duplication. Now come to the deletion. The deletion in which one codon lasts. Consider this chromosome. After mutation due to XYZ region, the D lasts, and the new structure having C and E. This type of mutation is known as deletion. The second one is insertion. Consider a chromosome which having A, B, and C. After mutation, another B is added to the sequence. This type of structural mutation is known as insertion. The fourth one is the translocation. In translocation, consider the two non-homologous chromosomes come close to each other. After close to each other, for example, the A transfer from this one chromosome to this one, and the new chromosome after mutation having A, C, D, E, D. This type of mutation is called translocation. That one codon transfer to another one. Duplication, consider a chromosome, this one, if the chromosome having A, B, C. The duplication occur in B. Consider this new one after mutation having A, B, C, B. The B are double. This type of mutation is called duplication. Now, on the basis of change in number and chromosome number change, they have two main types. The first one is Aneuploidy. An Aneuploidy increase occur or decrease occur in whole number. And whole number of chromosome decrease or increase occur. Consider they have four types. The first one is the monosomy, the second one is the nolisomy, the third one is the trisomy, and the fourth one is the tetrasomy. In monosomy, and the whole set one decrease. Consider a human have 46 chromosomes. The one chromosome become large and become 45. This type of mutation is called monosomy. And nolisomy, and the whole number 2 decrease occur. Consider the human case again. Human have 46 chromosomes. The 2 chromosome large, it become 44. And this type of, in this condition, we called Aneuploidy. And the third one is the trisomy, and trisomy and the whole set one increase occur. If we consider human have 46 chromosomes, one increase which become 47. The fourth one is the tetrasomy, in which increase occur and the two increase occur in the whole number. For example, the whole number in human is 46 plus 2, which is equal to 48, and the upstream at 48 is known as tetrasomic condition. The second type is Euploidy. Euploidy, a complete set of chromosomes increase. For example, they have three types. One is the triploidy, the second is tetraploidy, and the fourth one is polyploidy. And triploidy in the whole set, one set is increased. One complete set is increased. For example, if we consider 2n is 33 plus 33 in human case, one set 33 is increases. This type of condition is called triploidy. In tetraploidy, two sets are increased, and polyploidy, many sets are increased. Thanks for watching. For next video, please subscribe.